Okay. Mm. Okay, so first we're going to talk about where proteins are made. And in eukaryotic cells, proteins are made in the cytosol. And in the cytosol, there are ribosomes. And there are two types of ribosomes we talked about in class. There are free-floating ribosomes, which float around freely in the cytoplasm. And then there are membrane-bound ribosomes, which dock onto the endoplasmic reticulum. And that would be the rough ER. And proteins that are bound for the endomembrane system have to be made by ribosomes that are attached to the rough endoplasmic reticulum. And these ribosomes will latch onto the outside of the ER membrane, and they will help proteins to go into the ER, as well as the proteins that are secreted out of the cell and proteins that go to the lysosome and the Golgi bodies. So any protein that's going to go in the endomembrane system has to be synthesized by a membrane, has to be synthesized by a ribosome that is docked on the ER membrane. And then proteins that don't go through the endomembrane system will be made by ribosomes that are freely floating in the cytosol. So these proteins include mitochondrial proteins, nuclear proteins, and cytosolic proteins, as well as chloroplast proteins. But I don't think we'll be talking about chloroplast proteins for the purpose of this class. Oh, does anyone have any questions on anything I just said? No? Okay. And so this is just a diagram of what I just said, of how you have the two types of ribosomes, so the membrane-bound ribosomes and then the free ribosomes. And so the freely floating ribosomes produce proteins that end up in those locations, and then the membrane-bound ribosomes make proteins that enter the endomembrane system. I see notifications in the chat, but I can't read the chat, Rachel, so can you tell me if there are any questions?
Yeah, it's just about check-in process. You're good.
Oh, okay. Thank you. Okay, so Rachel, do you want to do this next slide?
Yeah. So we talked about the signal sequences you need to go through the endomembrane system. I would help to know what the endomembrane system is. It's a group of membrane-bound organelles. They work to package and transport lipids and proteins. I remember in middle school biology, we talked about the Golgi body as kind of like the UPS system of the cell. So you have your ER, your Golgi bodies, your lysosome, and the cell membrane. That's the order it goes through. If you need to get to the Golgis, you have to go through the ER first. There's no skipping. But I think we're going to talk about, yeah, that's later in the slide. So this is also called the secretory pathway. Any protein that functions in any of them must travel this way, like I just said. So the movement of the proteins through the endomembrane system starts in the ER. You need that localization sequence like we talked about in class, but then they kind of move off if they're going further. So say you have a protein that has a sequence for the Golgis, it'll go to the Golgis or the lysosome, but it has to first go through the ER. So always remember that you can't just jump from the cytoplasm to the Golgis, you have to go through the ER first. And then also what Laura really wants you guys to remember is that if you have an ER localization sequence and you need to go to the ER, you need a second sequence on your protein called the retention sequence. Otherwise, your protein will continue to go through the secretory pathway until it is in the membrane, because the membrane's the last stop of the pathway. So you need that retention sequence on your C terminus of the protein if the protein's final destination is to stay in the ER. Does that make sense? Does anyone have any questions? We have a visual on the next slide we can go to. But if anyone has any questions, please just let us know. No? Okay. I think we can go to the visual. In the book, it was saying proteins are tagged with sugar. What do you mean with sugar? Like glucose molecule it's tagged with? Michelle, someone asked,
Chat?
yeah.
Okay. Could you just reiterate the question?
It says in the book it was saying some proteins are tagged with sugar.
I'm not
Like
sure what they mean by sugar, but we're going to talk about different protein processes and how they get trafficked, but there's signal recognition particle for the ER proteins, and then nuclear import has its own separate function. So I think they
yeah,
might be talking about how the signal sequence is a binding site, maybe.
we keep cutting out. Oh, are we cutting out for other people? Can you guys hear us okay?
Can people hear what I said? Do they want me to say it again?
I think it's fine for most people.
Okay, just so you guys know, I have clear audio from my laptop, so when it's uploaded to YouTube, you won't hear the cutting out. It will come out clearly.
Yeah,
Yeah.
yeah. So, um, yeah, in terms of the glycoprotein, I don't really think we,
I don't think she's gonna cover the
yeah,
glycoprotein. I don't think that's something you need to know.
yeah, that's
If
what
it's
I
from the book, she doesn't really pick out much things from the book. The book is just a way to
know.
supplement the learning. Yeah.
Yeah, yeah.
All the material that you need to know is from her lectures, if that helps.
I think we can
Okay.
go on to the visual. Yeah, that
Yeah.
works. So yeah, here's your visual. Will these slides be available on Moodle? Are these slides available on Moodle, Michelle? Or do we just?
So the way these slides work, after the review session, I post an email within an hour of the slides, and then once the video uploads to YouTube, I also send the link out for that. So it's not through Moodle, it's through your Gmail, your UMass email.
Yeah. So here's the visual we have. Basically, the endomembrane system. You have your nucleus where they, get transcribed and then translation, blah blah blah. So you have your rough ER, and remember that the ribosomes that are on the rough ER are what are translating your proteins. Yep, where Michelle's pointing to, those are where your proteins will be translated if they're going through the endomembrane system. If they are not, they will be translated by the freely floating ribosomes that you can see kind of just around in the cytoplasm. This is just a good, it's basically just a good visual. It gives you a little bit about what the Golgi does, modifies lipids and proteins from the ER, sorts them, packages them. Then you have your secretory vesicles that from the Golgi that go out into the membrane, which is your last stop. But yeah, this is basically just the visual of what we just talked about. I know personally I like to see visuals, they help me learn, but if you guys have any questions, just let us know, either in the chat, because I can see the chat, and I'll just let Michelle know, because I know she can't see it when she shares her screen.
Okay, so for this one, I just wanted to point out that a protein, its default mechanism, if it just has ER localization signal sequence, it's just going to go straight through the ER, then it will pop off here at the secretory vesicles, and then from there, it will travel through the Golgi and onto another secretory vesicle until that vesicle comes into contact with the cell membrane, and then the protein gets secreted out of the cell. So that's if you have a protein that just has ER localization and there's no ER retention or a second sequence that retains it in the lysosome or anything like that. So that's what the image is showing you with the endomembrane system. And it's hard to see because it's kind of upside down since the ER is down here and the exiting the cell is up there. But that's the idea behind this image. Does anybody have any questions? I can't tell because I can't see the chat, unfortunately.
You're good right now.
Okay, so I will continue then. How do proteins know where to go? This is all due to signal sequences. So signal sequences are usually embedded in the amino acid sequence of a protein. In most cases, we're going to discuss cases where this isn't true. But try to imagine a signal sequence as a zip code. It will tell the protein where it's going to be living in the cell. And signal sequences, since they are part of the amino acid sequence, they cannot be added after a protein is made, which is separate from post-translational modifications, which we looked at a few lectures ago. And anytime that you have a protein that is localized to the ER, it must have an ER signal sequence or ER localization sequence. And this is because the ER signal sequence is recognized by a particle called SRP, which stands for signal recognition particle, which will bind to this newly made protein and ship it off to the ER. And if the protein does not have an ER signal sequence, then it's not going to be able to leave the cytosol, and it will stay in the cytosol after it has been translated. So that's kind of like if the protein does not have a sequence at all. And any protein that has to go outside the cell, like we said before, must travel through the endomembrane system. And once they leave the ER, proteins rarely come back out again. Does anybody have any questions regarding this slide? No? I'm going to take that as a no.
You're good.
Okay. And so I just want to shout out Tiffany from Discord because she found this great image, which I used for Bio 151. But it will basically, it's a flow chart of signal sequences. So you can see here, if a protein has ER signal sequence, the ribosome will dock to the ER, protein goes through the ER. And then if it doesn't have any of the other sequences, eventually a protein will end up secreted outside the cell. And then we're also going to talk about cases where if it doesn't have signal sequence and has any of the other sequences, it can end up somewhere else. So this is just a really good visual to help with signal sequences and figuring out where a protein could end up, depending on what sequence it has. Anyone have any questions on this? No?
Actually, I do have one.
Okay.
So in the middle of the diagram there, it says signal anchor sequence.
Mm-hmm.
Why is it that if there is a signal anchor sequence and there isn't, it still ends up in the same place?
Um,
Does that make obsolete?
no, so signal anchor sequence is when you have, signal anchor sequence talks about when you have a transmembrane protein. So this is a protein where part of it is on the exterior surface of a membrane, and the other half of it sits inside the membrane. So it's kind of like part of it sticks. So if the protein was a transmembrane protein, it's like in the middle of the membrane. So it's like stuck in the middle, and part of it, the top part, is above the ER, and then the other part is like in the ER lumen. We won't really talk much about the signal anchor sequence for this class, but I just found the diagram just helpful to figure out for all the other ones we do talk about. But yeah,
Okay.
the signal anchor sequence talks about a transmembrane protein, which is kind of like when you have, like, if a protein was like a cell receptor, the receptor part would be at the top, and then on the inside of the ER would be the rest of the protein, which is a bit complicated, and I don't think Professor Francis gets into that for this class.
Yeah, she definitely does not expect you to know about the signal anchor sequence, but this flow chart just kind of gives you a visual of where things end up.
Right, so most likely in this class, when you look at this flow chart, you're going to say, yes, there's a signal sequence, but there isn't an anchor sequence. So then you continue on this way, and so then the protein would not have would have a signal sequence in the ER, then it would not have a signal anchor sequence. So then it goes through the ER lumen, which is like the body of the ER, like the middle of the ER. So it keeps going through the ER, then it will go to the Golgi on the vesicles. And then eventually your protein will either stay in the Golgi, go to the lysosome, or go outside the cell. So I hope that makes a bit more sense in terms of how you look at this flow chart.
Oh yeah, this was really helpful. Thank you.
Yeah, no problem. Okay, cool. So I can continue?
You're good.
Okay, so yeah, I'm gonna do this one as well, and then Rachel can do the next one. Um, so signal sequences, like we said, target proteins towards their final destination. And a huge emphasis for signal sequences that Professor Francis likes to mention a lot is that signal sequences act as binding sites for another protein. And so you see here in this image on the right, we have, I want the pointer thing. We have this newly made protein that needs to go into the nucleus. And so the signal sequence, which is located on the exterior surface of the protein, is recognized by another protein called importin. And importin will bind to that and bring the protein through the nuclear pore complex into the nucleus. This blue space is the nucleus. And so that's what we mean when we say signal sequences can act as binding sites for other proteins. And for the most part, proteins enter organelles unfolded. And the only exception to this rule is when we look at proteins that are trafficked to the nucleus. Proteins that enter the nucleus are fully folded and fully made. And this is because the nuclear pores inside the nucleus are able to squeeze the protein through. And this process is heavily regulated. And N-terminal sequences are normally cleaved when they enter the ER in mitochondria so that it does not exit the organelle. And we're going to look at some images of that, so hopefully that will make a bit more sense later on. Does anybody have any questions?
We're gonna just keep asking you guys if you have any questions at the end of each slide, so we don't have to search for them at the end of the presentation.
Yeah, so feel free to jump in either by chat or by voice if you have questions.
Yes.
I'm going to assume there's no questions if I don't hear
Yeah, I
any.
think you're good.
Okay, Rachel, you can do this one.
So here's another visual, basically just where they end up. So Professor Francis mentioned co-translational transport. Basically, this is just that your protein is being translated while also being translocated into the ER. So that's what we mean by those ribosomes that are embedded in the ER membrane are what are translating your proteins that will enter this endomembrane system. And then you see as it after that, you go through the ER. If you don't have an ER retention sequence on your C-terminus, you'll continue to the Golgi, which is that vesicle transport. And the other diagram, the big diagram we showed you of the cell earlier on. And then your Golgi will either send your protein to secretion in the cell exterior through the vesicles, the plasma membrane, the lysosome, or other parts of the endomembrane system, which you don't need to know for this class. The main takeaway here is that you need to have a retention sequence for the ER if you're going to stay there. If not, you'll go through this whole system until you end up on the outside of the cell. And then you have your proteins in the cytosol that end up in the mitochondria, the peroxisome, nucleus, chloroplast. So in the mitochondria, the protein is translated before. Correct. Yes. So the protein is translated before it is transported there because it's translated by freely floating ribosomes in the cytosol. Then it will be transported to its final destination, whether it be the nucleus or the mitochondria. Those are the two she mainly focuses on, not really the peroxisome or the chloroplast. But that's the main difference between the two. You have this transport while you're being translated and then transport after you're being translated.
Yeah, and there will be a I made a chart with similarities and differences.
Yep.
To help you summarize that.
I think that's the next slide.
Yeah. Oh, it is. Yeah, so basically this is everything that we have said before. You'll notice some similarities and differences here that for your ER and your mitochondria, the signal sequence is cleaved as soon as the protein enters that specific organelle. And then for the nucleus, the NLS sequence stays on the protein at all times. And when the protein enters the nucleus, it is fully folded and fully made. Whereas when the protein enters the ER and the mitochondria, it is being pulled in while it's being synthesized. So it kind of is like enters unraveled,
Mhm.
kind of like spaghetti-ish, sort of. There's some diagram of it that I have. But yeah, so in the ER and mitochondria, your protein enters unfolded, and then once it's inside the organelle, the signal sequence is cleaved, and then you have chaperone proteins that are inside each organelle ready to help the protein refold. And yeah, the nucleus, the protein enters fully folded and fully made because it can squeeze through the nuclear pore complex. And then another key difference I wanted to say was for the nucleus, your signal sequence is not found in the primary structure, but it's part of the folded protein. So it will be located on the outer surface of the protein. And then in your ER and mitochondria, it's embedded into the amino acid sequence, and they're normally located in the N terminus. And these three are the main organelles she focuses on in class, I'm pretty sure.
Yeah.
Those are the main ones. So no peroxisome really, and then I don't think she does. Yeah, so no peroxisome, no chloroplast. Yeah. So does anyone have any questions on the summary I just said? No? Okay. And yeah, these are just diagrams again I found from Discord. And it just shows you the how in the mitochondria, while the protein's being synthesized, is being carried over to the mitochondria, and then you have the chaperone proteins that are in there helping it refold, and then the signal is cleaved in there. And yeah, and then you also have the ER import there too.
Think of the protein when going into the ER, the mitochondria, when it's unfolded, as each individual bead of amino acid going in once at a time. That's what we mean on the chart by it goes in when unfolded, it's by primary structure, basically.
Yeah. Yeah, so does anyone have questions about that? These are just some visuals I thought were interesting.
Could you repeat again what you meant, like unfolded primary structure? Like, is a protein that's considered unfolded in its primary structure because tertiary is when it's folded?
No, so when I say protein enters unfolded, so maybe it helps if I show the nucleus.
Yeah.
So in the nuclear pore complex, when it's going to the nucleus, this particular protein, which is this golden thing, it does not unravel, and it does not unravel and have show each of the individual amino acids. So that whole thing is like a chunk of protein. It does not unfold at all or unravel or enter in a like a strand the way it does in the ER and mitochondria. So the protein has to get fully folded and fully created, synthesized in the cytoplasm first before it is recognized by important and pulled into the nucleus. Whereas in the other one, in the ER, uh, the protein, while it's being synthesized, the N-terminus signal sequence is recognized by SRP, the signal recognition particle, and then from that point, it is dragged to the ER, where it is this way, where it's threaded through while it's being made. So it's in the process of being made, but it's already being pulled through. Does that make more sense?
Yeah, that makes sense. It's just I don't know. I thought I heard something about like structure, like primary structure.
Well, what I meant by that is what you see when you're translating a protein, it's coming out in its primary structure. So you have each individual amino acids being translated into the protein one by one. That's what it means by this co-translational transport. Um, that's basically what it means, because you're not folded yet, because you're still making that protein and it's going through the same time it's being translated.
Okay,
Does that
so like
does
yeah,
that make
that makes
sense?
sense. So like the ER and the mitochondria, they would be translated in their primary structure while they're being translocated to the like wherever their transportation location is, whereas like the nuclear proteins, those would be a tertiary structure because it's already folded.
Yeah.
Almost.
Well,
Well, so you wouldn't be transported the same time you're being translated for the mitochondria, but yes, for the ER,
Okay.
um, the mitochondria you're already translated, and then it will be unfolded before it enters the mitochondria. For the ER, what you said is correct. It will be translated while it's being translocated into the ER in that primary structure, and the
Okay.
nucleus would be tertiary. But yeah.
Okay, thank you.
yeah,
Yeah.
I also want to bring up another point too. The reason why I phrased it as um, primary structure, because what I wanted to emphasize is that signal sequence is not something where you create a protein and then you just tack it onto the end. Signal sequence is already built into the protein itself within that sequence of amino acids. So for the ER and the mitochondria, you're going to find your signal sequence, which is already built into the protein. It's part of the sequence of amino acids. So it's towards the beginning of the protein polypeptide chain. You will find in the N terminus that signal sequence. And that particular sequence is recognized by SRP in the case of the ER. And so that's what I mean by it's embedded into the primary structure. And so then for the nucleus, it's not embedded into the primary structure, but it's part of the protein. It's not added after. It's just part of the protein, the folded structure. So then it's on the exterior surface of the protein, if that makes sense.
Yeah, so then what about the mitochondria then? If it's the signal sequence embedded into primary structure, like it just means that like as it's being translated, it's embedded, they're both on the N terminus. That's something you're going to see happen. But as far as its translocation, like Rachel said, the mitochondria is going to fold, but then unfold before entering into the mitochondria. Where chaperone proteins will help it refold or bring it into the mitochondria, and then it'll fold on its own.
So yeah, so in this diagram, it looks like that while the mitochondrial protein is being made, there's a protein that recognizes the N-terminus signal sequence and pulls it towards the mitochondria, and then it gets threaded through there. And then once it's on the inside, that sequence is cleaved off, and then it enters the mitochondria unfolded. Then once it's inside there and it's safe and it can't go back out of the mitochondria, the signal sequence is cleaved, and then it is properly folded into its tertiary structure.
Yeah.
That makes sense.
So that's when those chaperone proteins come inside the mitochondria to help it refold.
Whereas the nucleus, the entire time, the protein does not get unraveled. It doesn't get threaded through by the sequence of amino acids.
It's just the whole clump of
the whole
protein.
entire cluster of the protein doesn't change at all. It just gets pushed through the pore complex.
Does that make sense?
Yeah, that makes sense. Thank you.
Yep.
Yeah, no problem. I forgot whose turn it is.
I think it was, you did the chart, so I can do this one. So yeah, these are just the type of signal sequences. We also have the chart on the next slide that she went over in class. I know the chart can be kind of scary when you first look at it. But basically, in terms of the signal sequences, so like Michelle said, they are embedded into the primary structure for the ER and the mitochondria, so it's not something added on. So for ER import, so this is to enter the endomembrane system, the N-terminus is hydrophobic amino acids followed by interspersed charged R groups. So you will have the first beginning part of it is going to be a big chunk of hydrophobic amino acids, and then you're going to have those charged R groups following it, which you'll see better on the next slide with like the different amino acids used as an example. The SRP, like we mentioned before, is what recognizes the sequence when it first gets translated out of the ribosome and brings it into the ER. And then for the ER retention sequence, you need this to stay in the ER. So in order to, if you say you have a protein and it's like my final destination is the ER, okay, you need a signal import sequence which is that hydrophobic amino acid residues, and then you also need a retention sequence. Otherwise, the protein's just going to keep going through the pathway. So that ER retention signal sequence is at the C terminus, which is also, we talk about what the properties of those amino acids are in the next slide. But if you don't have that C terminus, because she loves to ask questions about this, then it will keep going through the pathway. In terms of the mitochondria, you're gonna have a lot of interspersed positively charged R groups. So your positively charged R groups are usually your basic amino acids. That's basically all you have to know. They're just interspersed throughout the signal sequence. For the nucleus, we're going to talk more about import and export into the nucleus a little later on. But you need this NLS, nuclear localization signal sequence, to enter the nucleus, and you need an NES, nuclear export signal sequence, to leave the nucleus. These don't get cleaved off, and the signal sequence is on the exterior of the protein, so it's not embedded like the ER and the mitochondria. And then we have this trick at the bottom. If you're going to the cytosol as your final destination, you will not have a signal sequence. So any protein that does not have any signal sequence will stay in the cytosol. Does that make sense? Okay.
So I just have a question about the ER retention. When you say must have import to ER sequence, are you referring to like the N terminus, like
Yes.
signal sequence?
Yeah.
And then for the retention sequence, like on the C terminus, that has to stay on, so that won't get cleaved, correct?
Correct.
Okay.
Because if you cleave it, then the protein's going to be like, wait, where am I supposed to go?
Yeah, so once the protein enters the ER, remember that that sequence ends up being cleaved. And then once it's safely into the ER and it can't go back out again, that signal sequence becomes cleaved. Then you read the rest of the polypeptide sequence and you see, oh, at the C terminus, there's ER retention. So once it's inside the ER, it can't get back out, but it can continue through the endomembrane system. And so the ER retention sequence is just kind of like a stop sign that says, hey, you should stay here in the ER, don't go anywhere else.
Yeah.
That makes sense.
Yeah, that makes sense. Thank you.
Yeah, sorry. That's why I have it for if the protein is going to live and work in the ER. So if it's going to stay there in the ER, it needs both sequences.
Mm-hmm.
And also it helps to think about it in reverse too. So if the protein just has ER retention sequence, but doesn't have ER import, there's no way it's going to be able to stay in the ER if it can't get into the ER in the first place. So it needs to have both.
Yep.
Cool.
Does that make sense, guys? Any questions? Okay. So here's that chart that Professor Francis showed you guys in class. It's just basically, she wants you to remember too that because the import into the ER, so this is just an example, you have all these hydrophobic amino acids in the beginning. It doesn't have to be methionine, serine, phenylalanine. It can be any hydrophobic amino acid. She wants you to remember the chemistry of it. So it's not usually specific amino acids that make these signal sequences, it's just the chemistry of it. So for the retention in the lumen of the ER, you have a basic amino acid, and then you have your negatively charged amino acids. If she gave you hydrophobic amino acids in this retention C terminus sequence, it wouldn't stay in the ER because it doesn't have the proper chemistry. So it doesn't have to be exactly the amino acids you see here. You just have to understand the chemistry behind the ones that are in these sequences, if that makes sense.
Yeah, so you just have to understand that the chemistry plays an important role in the function. So if you tweak a couple of amino acids for import into the nucleus, and you make all of the ones marked in pink for import in the nucleus, if you made them all blue, then there might be a chance that that protein will never make it into the nucleus because, like we said, the signal sequence is a binding site for another protein. So important would not be able to recognize that it needs to take that protein into the nucleus because of the difference in chemistry of the sequence. So does anyone have any questions? No?
I think we're good.
Okay. Oh, so I put in here a practice question, which will represent the types of questions she could ask on exam or problem set. So you guys can give this a try and see how it goes. Okay, so transmembrane proteins must have an ER import sequence, signal sequence on their N-terminus. Predict where you would find a membrane receptor protein if you engineered its gene such that you remove the ER signal sequence. Assuming that your receptor protein does not completely misfold or get degraded, where would you find your receptor? And membrane receptor protein is just another word for transmembrane protein. So does anybody want to put in the chat, or do you want to say out loud what you think, where you think the protein would end up?
We got one cytosol.
Anyone else?
I also want to guess. Take a
I'd
shot.
agree cytosol too, just because if you don't have an ER signal sequence, it's not going to be able to like go into the transmembrane or plasma membrane because it has to go through the ER in order to get to that. So it'd probably just end up in the cytosol.
Yeah, we have a few other cytosols in the chat too.
Nice, so you guys are all spot on. So you'd expect to find this protein in the cytosol. And this is because, just like Jesse said, the receptor needs the ER signal sequence so that the signal recognition particle can recognize that, and then it can bring it into the ER. And then there's the whole separate transmembrane protein sequence that we don't talk about in class. But for the sake of this question, you just need to know that it needs the ER signal sequence to get into the ER. And yeah, so without the ER signal sequence or any other kind of signal sequence, the receptor would just end up floating in the cytosol. And then I put here an analogy to sort of help with this question, because why not? So just imagine yourself getting an Uber ride, and if you don't tell the driver your location, and I put for this example using GPS signaling, then they'll never be able to find you and take you to your destination. And so this is the same way with the signal sequence. So if there's no ER signal sequence on a newly made protein, then there's no way for the signal recognition particle to recognize that this protein needs to go to the ER and bring it to its final destination. So yeah, that's just a sample question that she asked me actually when I took the class.
She loves to ask questions like, what would happen if you tweaked this signal sequence or changed this part of the pathway? What would happen? She loves asking those types of questions that make you think about, you have to know the original function and then what would happen if you changed it. So like this question is a great example of that. And then another one could be, I know she loved to ask when I took her class, like, you had a bunch of polar amino acids in the ER signal sequence, where would the protein go? And it wouldn't, because it technically wouldn't count as an ER signal sequence because they're not hydrophobic, if that makes sense. But that's just another example of the types of questions she just likes to ask.
But yeah, so basically this is a good question because it just shows, do you understand the basic mechanism of signal sequencing? And then would you understand what happens if you make a change? What could possibly happen? Yeah. So yeah, I'm glad most of you said cytosol though. That shows that you guys are understanding it pretty well. So does anybody have any questions about that question? No? Okay, I'm gonna keep going. And the transmembrane protein I chatted about in this question, these are real proteins that we will get to eventually when we discuss pathways. And so I just put this example here. I'm not going to tell you what it's called because you don't need to really know that now, but soon.
We'll get to it later on in the semester.
Yeah. So as you can see, it's kind of just like, well, this is a transmembrane for cytosol, but pretty much same idea. We have part of the protein sticking out and then part of it inside the cytosol. But yeah, we will get to it. And so, yeah, Rachel, do you want to do the next part?
Sure. All right, so now we have some secretory pathways, just some real life examples of protein trafficking, like hormone secretion. This specific example is insulin, where insulin signaling. So the hormone's final destinations from the cell, I don't know how much about hormones you guys know from either other classes or whatever, but most of them end up into the bloodstream or target organs, wherever they're going. So this is just an example of the real life protein sorting and trafficking that occurs in our bodies every day. So this particular hormone, I think it's insulin. I don't think it says though, is going through the endomembrane system. So basically the mRNA binds onto the ER surface ribosome and gets translates the protein. You can see it down at the little number 1, this little blue red squiggly line. That's that protein being translated while also being put into the ER, like we said, for that endomembrane system. And then once it's in the ER, that signal sequence gets chopped off. As you can see this little red squiggle down at number 2, that's the signal sequence being chopped off. And you have this prohormone that doesn't have an ER retention sequence on its C-terminus, so there's no stop sign, so it keeps going through those vesicles, through the Golgi. The Golgi then sends it on to more secretory vesicles and then out through the membrane. And then this is basically where the hormone moves into the target organ or the circulation because it gets chopped into pieces from this release signal. And that's basically just your example of a real life protein trafficking and sorting, about how this hormone ends up from the cytosol through the endomembrane system and into the circulation. Anyone have any questions on that?
Yeah, I wanted to add something. So I don't know if the way I phrased it earlier didn't make if it was difficult to understand. But for ER trafficking, simultaneously, so at the same time as translation is happening, that protein is being pulled through into the ER, which is completely different from when we look at nuclear localization, because the protein at that point gets fully translated in a cytosol and then it's moved to the nucleus. Whereas when you see an ER in mitochondria, while the protein is still being synthesized, it's being pulled through the ER. So yeah, I hope that makes just a bit more sense than what I said earlier. Cool. So I'm going to move on. And so this is another real life example I think she went through in class. It's just a lysosomal enzyme. So it's pretty much the same thing that Rachel said. And as you can see, it travels through the endomembrane system. Excuse me. And yeah, that's just another example of protein sorting and trafficking. And yeah, enzyme is a protein, so I'm just showing you another example of that. Cool. Any questions? Okay. Rachel, do you want to do?
Yeah. So now we have our nuclear import and export. She loves to talk about nuclear import and export. I know we talked about it a little bit in class today, but basically you have this GTPA superfamily, and the example we used in class is RAN, which is a part of the RAS family of GTPases. You don't really need to know the details of it. You don't need to memorize any of this, G protein superfamily. Just know that RAN is a member of the GTPase superfamily, and GTPases hydrolyze GTP, like a ATPase hydrolyzes ATP. It's an enzyme, so it's also a protein. That's basically what we want you to take away from these slides, is just that is what RAN is, because you'll hear in class say RAN, RAS, GTPase. This RAN is a part of RAS, which is a GTPase.
Yeah, so RAN is a protein, RAN is an enzyme, RAN is a GTPase. So RAN is all three of those things. And don't waste time memorizing the superfamily. You do not need it. This is just one of those kind of fascinating things to know. And yeah, it's just to prepare you because we're going to talk about RAS soon, so not to get those ones confused. But again, RAN is an enzyme, RAN is a GTPase, RAN is a protein. So yeah, any questions about this? No? Okay.
I can do this slide if you want.
Yeah, okay, you can do it.
I spent a lot of time studying these. So GEFs and GAPs are what you heard her talk about in class too. We have the diagram, which we'll show later on, the same one from class and your textbook, I'm pretty sure. But GEFs and GAPs are proteins that regulate the RAS family and GTPases. So remember, in this case, talking about RAN. You have your GEF, which is your guanine nucleotide exchange factor. This is found in the nucleus, and GEF's job is to bind to RAN, change its affinity for GDP so that RAN can bind to GTP. So your end result from GEF is you're having your RAN GTPase bound to GTP. So it's assisting in RAN GTPase in dissociating away from that GDP because it's decreasing its affinity for GDP. And there's a lot of GTP around in the nucleus that can easily slip in and bind to that active site. And once RAN is bound to GTP, it is now active. So your GEF is making your RAN GTPase active, and its job occurs in the nucleus. And your end result is that it's bound to GTP. Then you have your GAP, which is your GTPase activating protein. So Professor Francis mentioned this in class, but your GTPase can hydrolyze GTP on its own. It has that ability. That's why it's called a GTPase. But it does it very slowly. So GAP increases this rate. So it basically just catalyzes the reaction, makes it quicker. And the end result is that RAN GTPase is now inactive because it is now bound to GDP, not GTP. And you'll only find your GAP in the cytosol because that's where it does its job, and that's where RAN GTPase needs to hydrolyze GTP to GDP. The RAS movie in the movies folder shows a really good visual of this. And basically to help GTPase hydrolyze this GTP, GAP sticks an arginine into the active site of the GTPase, which increases the rate of the reaction. She mentioned it in class, that's why I'm mentioning it now. I don't know if she'll ask you the specifics about that, but she may want you to know that arginine is a basic amino acid. And that's why she may ask, like, I don't know, like if you put another type of amino acid in, like, will it do its job? I don't know if she will ask that, but she mentioned it in class, so I felt it was enough to mention it now. But we're going to go into like more detail about these with the diagram for each nuclear import and export. But does anyone have any questions on these for now?
I wanted to add something real quick.
Mhm.
I don't believe, at least for me, when I took the class, that she asked question about the arginine finger, but she might. Is something to know. And I really want to stress this important point because a lot of times we see this when we look at problem sets and exams, but students will say that the GAP is the one that can hydrolyze GTP to GDP, and this is incorrect. So just like Rachel said, RAN is an enzyme and RAN is a GTPase. So because RAN has its own intrinsic ability to hydrolyze GTP to GDP, but it does it at a very slow rate. So this is why the GAP is used to help RAN hydrolyze GTP to GDP, but RAN GAP is actually GAP will accelerate the hydrolysis of GTP to GDP. So it makes it occur much quicker. But it's not the one doing the hydrolyzing, if that makes sense.
Right. So someone asked, are both of these considered enzymes?
RAN is definitely an enzyme.
RAN is an enzyme. GEF and GAP, I don't think they are considered to be enzymes. I know it may sound like it with GAP because it's speeding up this reaction, but I think she just wants you to know that they are proteins. They just regulate the RAS family. I'm not entirely sure though if she wants you to classify them as enzymes. I don't think she does.
Yeah, I don't believe these are enzymes, guys. They just help out in the process of nuclear import and export, but they themselves are not enzymes. The main shining star that is the enzyme in the nuclear import and export is RAN, because RAN has its own intrinsic GTPase activity. So RAN by itself can swap out a GTP for GDP,
Right.
hydrolyze it. But I will double check on that and send an email.
Yeah, I agree. I think it's just that the RAN is the enzyme because it has that hydrolyzing ability. And GEF and GAP are really just there for support, basically.
Yeah. Okay, so that's all the questions. We can go to the next slide.
Yep.
I think that's just the same thing as we said
Yeah,
before.
it's just more in detail about what GEF does. It just causes this change in conformation so that RAN loses that affinity for GDP. You're not converting GDP to GTP, you're switching out one molecule for the other. I know that's sometimes a misconception some students have is they think GDP is being changed to GTP, but it's two different molecules.
Yeah, so what you're doing, what's going on is that the GEF will decrease the affinity that the RAN enzyme has for GDP. So then when it's inside the nucleus, because there are more GTP molecules that are abundant in there, the GTP, once it sees the empty binding pocket in RAN, it will just kind of diffuse in there. So yeah, the GEF and the GAP sort of help to assist RAN in changing its conformation, and that in turn that will tweak the affinity that it has for GDP or GTP.
And where it says GEF dissociates, so basically GEF just binds to RAN. So that's why it's not it has to bind in order to do all this. So once it's bound to GTP, sorry, GEF dissociates from it, and because it's no longer needed.
Yeah. And I will address this when we look at the actual cycle of nuclear import.
Yeah.
It will make more sense then.
Yeah, it's easier to see.
Okay, so nuclear import. So I'm going to try and explain this as best as I can. So basically, maybe I'll use the pointer. So you start off in the cytosol. You have important. Important is a protein, just a protein. Important is going to be looking for the nuclear localization sequence that will be present on the exterior surface of a newly made protein. And it looks for the signal sequence, the NLS, and it will bind there because, as we learned, signal sequences work as binding sites for other proteins. Then important and the newly synthesized protein will be stuck together. And this is good because we want important to bring this protein inside the nucleus. Then I gotta move something. Then once it's in the nucleus, where did my pointer go? I can't see. Anyways, once it is in the nucleus, important, and this protein enters the nucleus, then it will RAN GTP, which is the activated RAN, will bind to important, and this causes important to release the protein that is delivered to the nucleus. And then once this happens, the protein that needs to go into the nucleus has entered the nucleus, and that's successful. But then we need to bring important back out into the cytosol so it can do it again for the next protein. So then your RAN GTP, which is attached to important at this point, will leave the nucleus and go to the cytosol. And then in the cytosol, the GAP will assist RAN, or accelerate the hydrolysis of GTP to GDP. And so when RAN is bound to GDP, it has it decreases the affinity that it has for important. So when RAN is bound to GDP, it's able to let go of important. And so important is able to float freely in the cytosol and do it all again. So does anyone have any questions about what I explained?
NPCs, someone asked what NPC is. That's just the nuclear pore complex.
Yeah.
Yeah. I also like to think of important kind of like, I guess you could think of exportin too, but I kind of like to think of it as the PVTA. You know, you're the cargo protein, you need to get on the bus, you get on the import in, it takes you where it needs to go, then you need to get off at your stop. You don't want to stay on. So then you get off once you're in the nucleus and the PVTA goes back around and does its route. So that's how I sometimes like to picture it. Just remember, you need that important to get into the nucleus, just like if you're going really far, you need to get on the bus will take you where you need to go.
So I have a quick question. Since the cargo protein is the one that actually has the NLS sequence, which allows it to go into the nucleus, does important have an NLS sequence too? Because how would a cargo protein rely on important if it already has a localization sequence? Like, why couldn't it just do it itself if it has a localization sequence?
Well, for any protein that wants to go to the nucleus, this is the general way that it has to go through. It can't go by itself. It needs the help from important, because the signal sequence is a binding site, and then important will bind to the protein and bring it into the nucleus. That's true for any protein that goes to the nucleus.
So you can think of it kind of as like an unfinished puzzle, like you need the important to bind in order for this actual protein to work. Like, I guess kind of like that, if that makes sense.
I guess so, but the protein itself can't just enter the nucleus, because the nucleus has nuclear pores, and it can't just squeeze in proteins in there all the time, because proteins are kind of large. So the process is heavily regulated to bring in the proteins, which is why you need important to bring it in. It can't really just go there by itself.
That's also why important needs to be bound to RAN GTP in order to leave. It can't just go in and out on its own. Does that make sense? It needs that RAN GTP to take it out, and it needs the cargo protein to bring it in.
Yeah, that makes sense.
Yeah, so even the ER too, like the SRP recognizes the protein and recognizes the signal sequence and brings it in. The protein can't just enter ER by itself. It needs help. And the signal sequence offers the help, and then important can recognize it and bring it in. Hopefully that makes more sense a bit.
Yeah, that makes more sense.
Okay, cool. Are there any other questions regarding nuclear import? No? Okay. Oh. My screen always does that. Okay, I'm turning off the pointer. Okay. Rachel, you can do export if you like.
Yeah, sure. So export, you're still using your GEF and your GAP. The only difference really is that you're starting on the inside, you're starting in the nucleus, and you're trying to get out. So you have this NES instead of the NLS on your cargo protein. So your NES is your nuclear export sequence. And then the difference here also is that in order to get out of the nucleus, you need this cargo protein to not only bind to exportin, but exportin also needs to be bound to this active RAN GTPase. So you need this complex of basically 3 proteins, because RAN's an enzyme, and then you have these 2 more proteins as a complex of 3 in order to exit the nucleus into the cytosol. And then when you have your cargo released and your exportin released, that's from the help of GAP. So GAP does the same job it did in nuclear import. It just speeds up the hydrolyzation of GTP to GDP, which decreases RAN's affinity for exportin, and exportin will let go of the cargo protein before re-entering the nucleus to help bring out more cargo proteins. But you can see here on the inside of the nucleus, when you have GEF is doing the same job. It's binding to RAN to change its conformation, so its affinity for GDP decreases, and GTP can slide in and make its itself active with RAN, it's now active. And then once you have this RAN GTP active enzyme, it binds both exportin and the cargo protein. So you need this, that's the biggest difference, is just this complex of 3 will take it out of the nucleus. Does anyone have any questions? It's pretty similar to nuclear imports, just that difference there. No? Okay.
Cool. Yeah, so just like Rachel said, that's the main difference, is that you have that triple complex forming with the RANG GTP, the exportin, and the cargo protein. Whereas an importin is able to bring in the cargo protein into the nucleus alone without the help of RAN. Yeah, so any questions there?
That does bring us to the end of our presentation. So yeah, if you have any questions, let us know. You can ask us now, you can email us later if you think of them later. Yeah, you're welcome.
Yeah, and we also have some extra pictures of
yeah, we have some extra slides. We skipped them representing, so this was just a quick, can you see them, Michelle? Are they like there?
let me see.
These were just some of my notes. So I recently took the MCAT this summer. So these were just some of my notes from the MCAT that we covered with signal sequencing. Yeah, you guys, you're welcome.
But yeah, we will include those when I email out.
They will, yeah, they'll be in the email. They're just there to help you visualize. So when you see them in the presentation, like the recording or the email or whatever, it's there. It's just another visualization of where proteins end up, where they need to go. But don't worry too much if it doesn't